Hi, my name is Elena and I'm glad to see you on my YouTube channel. We continue our circle of lessons about tissues, and today I want to talk with you the types of epithelium tissue, their localization, functions and different properties. Let's begin with single layer epithelium. Depending on the cell's form, we can divide it into three types: squamous epithelium, cuboidal and columnar epithelium. Squamous epithelium have flat cells. It is localized in different places. For example, pulmonary epithelium in pneumocytes that build alveoli of the lungs, endothelium of blood vessels and heart. Inner surface of arteries and veins is covered with squamous epithelium. As for capillaries, they are made only of these type of cells. They have no additional layers. Also, there are endothelium of lymphoid vessels, inner surface of cornea, and peritoneum. The main function of squamous epithelium is exchange function. Most of all, it is localized in organs where nutrients exchange happens, like oxygen intake and carbon dioxide removal through the flat cells of alveoli and flat cells of capillaries. Cuboid epithelium is localized in glands, ducts, convoluted tubules of nephron, ovary cells, and some other examples. There is no specific function of this type of epithelium, so let's move to the next type. Columnar epithelium is common for our intestine surfaces: stomach, small intestine, and large intestine. And there is a specific trait of small intestine surface. It is microvilli. Don't confuse it with cilia that are typical for silvery epithelium of our respiratory tract. Microvilli are static structures that increase intestine surface, so we can enlarge absorption capacity. This is truly vital for consumption of different nutrients that we digest. So, next group of epithelium tissue are multilayer epithelium. There are two types: keratinizing and non-keratinizing one. Keratinizing epithelium is located in our skin. We'll discuss skin layers in another lesson, but now I want to tell you about some layers of epidermis, our keratinizing epithelium. Keratinizing epithelium consists of 5 layers. Basal cells are connected with basal membrane. This area is called stratum basale. The proteins providing the attachment were discussed in our lesson, so watch it if you haven't done it already. Cells of stratum basale are stem cells. They provide new epidermis cells and maintain their own stem property. So, over stratum basale, there is stratum spinosum. These cells have polygonal structure. They have something like spikes. They are produced by basal cells. So, a little stem cell in basal layer is dividing into 2 cells. One of them continue being stem cell and another one goes to differentiating. Higher we can see stratum granulosum. According to its name, it contains specific granules. These granules contain a protein keratohyalin. It has a lipid-like substance that maintains cell keratinization. And there are structures that contain keratohyalin molecules. Those are lamellar bodies. Lipids in lamellar bodies are released into the extracellular space through exocytosis to form a lipid barrier that prevent water loss from the body as well as entrance of foreign substances. By the way, in this layer we can observe tight contacts between cells. It is another 2 separate cell layers. Because of these tight contacts, nutrients from blood can't reach above allocated layers, so they die, and it is the purpose of our body. Next layer is stratum lucidium. We will see the specific traits of this layer on light microscope. The meaning of the layer is not so clear, but we can say that it is located only in thick skin on our palms and soles. It is easy to remember this layer is shining in a light microscope, and if you compare the skin on our palms and the back on our palms, we can see the difference. We'll discuss types of skin in another lesson. But now let's discuss the last layer. The outermost layer is stratum corneum. It consists of keratinocytes, dead cells that maintain the barrier between our body and the environment. It is also a barrier for water evaporation and pathogen penetration. And the next type of multi-layer epithelium is non-keratinizing epithelium. It is located in that places where we need defense from different dangers. For example, in our oral cavity, we always eat something spicy, bitter, cold, hot, and others. But we need a defense from so severe conditions. Of course, single-layer epithelium is not enough for such tough conditions. The same situation is with rectum and anus. Here too, various aggressive conditions are observed. Another example, the outer surface of the cornea. Again, we need defense from different mechanical damage. But apart from these typical tissues, we have some specific ones. First is transitional epithelium. It is typical for bladder and ureter. It is intended for organs that are capable for great stretching. The top layer of cells, umbrella cells, they are round, but while stretching they become flat, so that really maintain high level of organ stretching. And the last type of epithelium tissue is really confusing. It can seem like a multi-layer epithelium, but it is not. We mentioned it in the middle of the lesson. Ciliary epithelium. It is localized in respiratory tract. Unlike microvilli, cilia are dynamic structures. They always move the mucus that is secreted on the pulmonary epithelium. So this type of epithelium have specific characteristics. Cells have different height, and the in microscope we can see it like a couple of layers or more, but that's not true. Every cell is connected with the basement membrane. Apart from ciliary cells, there are goblet cells that produce mucate for protection against pathogens, dust, and foreign particles. They move their cilia to get rid of penetrating substances, and it is vital for our immunity. So, I hope that I gave you the first introduction with types of epithelium. Of course, the topic of epithelium tissue is more profound, but it will be better to talk about each specific type in connection with the organ and the function. So I hope that this lesson was useful for you, and if it was, please subscribe, leave comment, and I will wait for you in the next lesson. Bye bye.